I'm happy to introduce the third speaker of the morning session, Dr. Ravi Raju. He's an attending physician in the neonatal intensive care unit at Boston Children's Hospital, an instructor in pediatrics at Harvard Medical School, and a research scientist in my group at the Picower Institute for Learning and Memory. And his talk today is Dissecting the Neural Mechanisms Underlying Environmentally Induced Resilience. Ravi.
Yeah, thank you. Is the mic on?
Mm-hmm.
Yeah. Awesome. Thank you, Larry, for that introduction. It's such an amazing experience to see all of you guys in this audience, and we have, I think, almost 100 people online virtually this early in the morning to see that we care about this issue deeply, and I'm really excited to share our work with you on this topic. So, to me, one of the most interesting and frankly understudied questions in medicine and really society today is how do the social and environmental factors that we're raised in, that we inhabit, affect our health and well-being long term? A large body of epidemiological data to date suggests that in the United States, up to 70% of our total morbidity and mortality as a population can be accounted by being caused by these social factors, broadly known as the social determinants of health. We have overwhelming epidemiological data that this is a strong association, but what's lagging is a mechanistic understanding in why these exposures and why these environments are so detrimental to our health, and as Dr. Sege mentioned, why they can be so beneficial. Now, I know a couple of people have mentioned the Adverse Childhood Experiences Study. If you'll be patient with me, I'd really like to walk through this in a little bit more detail with you, because this, along with my clinical experiences in residency and fellowship, are the reason why I switched fields 7 years ago and decided to work under the mentorship of Li Wei Tsai. So in the early 90s, or probably late 80s, a team partnered up between the Centers of Disease Control and Kaiser Permanente, and they had a hunch that these early life experiences that children were encountering were causing long-term morbidity and mortality, but they wanted to prove it. So in a systematic way, they retrospectively and now prospectively chronicled and tried to kind of create different forms of exposure to adversity surveys to determine what the exposure to adversity a particular child might have had, and then look at their long-term health outcomes. And the studies, frankly, just floored even the actual investigators. What they found was that for almost every mental health disorder that they looked at, there was a significant increase in the risk of developing that disorder if you were exposed to more adversity. Sorry, I think I'm advancing instead of laser pointing. Exposed to adversity. So for example, if you take the example of depression, if you were exposed to 4 different types of adversity, you almost had a 60% chance of developing depression as an adulthood, representing a 3-fold increase in the risk over the baseline population. This was true for substance use disorder and anxiety. I think what astonished them even more was that when they looked outside of the mental health space and just took the top 10 leading causes of death, as Dr. Sege mentioned, and looked at the association with childhood adversity, they found that almost every single leading cause of death in the United States was significantly and positively associated with exposure to adversity. To me, it's shocking how you're 5 times more likely to develop something like Alzheimer's dementia in your 60s, 70s, and 80s based on exposures that you might not even have a conscious memory of. I think what elevates this to the sort of the level of a public health emergency is just how pervasive ACE exposure is in society. In the United States, it's estimated that at least almost 64% of children are exposed to one ACE, and over 20% of children are exposed to 3 or more ACEs, really highlighting that if we multiply this times this odds ratio, that a large percentage of morbidity and mortality in the United States today is actually attributable to these early childhood environments. Now, this is like really sobering and depressing news, but actually I think embedded in these studies is actually a silver lining. And that silver lining is if you can prevent the exposure to adversity, you can actually promote resilience and ensure that children grow up without risk of developing these disorders. And so we've actually known this for a while. It's almost like self-evident that positive supportive environments are going to be good. But people have studied it and shown this. This is almost a study that's 50 years old now out of Kansas City, where they looked at the vocabulary development or verbal development of infants in childhood, comparing those that were raised in an orphanage with those that were the children of professors at the local university. And what they found was that verbal development increased in children that came from the more higher SES backgrounds or socioeconomic backgrounds. This has gone on to be sort of coined in a famous term, the 30 million word gap, and it's really attributed to exposure. It's just that children from high socioeconomic backgrounds tend to hear on average 30 million more words in their first 5 years of life than children who come from lower SES backgrounds. We're starting to dissect the mechanisms that are at play here. So researchers have more recently showed that socioeconomic status can predict the rate of brain growth and brain development in the first 3 years of life. So here you can see the trajectories of growth in brain volume in the frontal cortex, stratified by which third of socioeconomic status you come from, suggesting that there are actually important mechanisms underlying these epidemiological observations. So positive environments are good, we know that. But also what's really cool is that we think that positive environments can rescue and mediate the effect of ACEs, as Dr. Sege beautifully showed with the PCE study. I think another study that really highlights this and is one of my favorite is the Bucharest Early Intervention Project. So with the collapse of the Soviet Union in the early 1990s, Western researchers started to go into Eastern Europe and found that, especially in Eastern European countries like Romania, that there were orphanages in which children were deprived of sensory experience largely for the first few years of life. Now, what they decided to do, simply because there was a limit to the number of eligible foster care families, they could ethically essentially randomize the children that were in these orphanages to receiving foster care or remaining institutionalized. What they found when they first looked at these orphans was that there was a shocking difference in DQ as developmental quotient or developmental index, which is a proxy for IQ that's done in early children, a shocking difference in the level of DQ between institutionalized children and then surrounding community children. What gave them hope, though, was in the group that was randomized to the foster care group at age 18, they found that the IQ was able to be rescued in the children that were foster cared compared to the institutionalized group, suggesting that these supportive environments can actually rescue the effect of early adversity. And so what I think that epidemiological body of work sets up for us is a nice conceptual framework where we know if we take on the y-axis, say for example cognitive function, which we've been talking about in terms of development, and age on the x-axis, that stressful exposures are going to impair your development and potentially cause a baseline hit that makes your sort of development impaired over the long term. But we know that positive and nurturing environments can actually create resilience and increase that baseline. And then there's the middle of the course where you might be exposed to adversity early, but if you have certain rescues, you might be able to improve your outcomes long term. And so really the question that has driven our work in this space is what are the mechanisms of how these important environments have such a beneficial impact? Now, I think we can say, like, it may seem self-evident that if you're exposed to positive environments that that's going to have a beneficial effect, but we know that there are actually changes in brain structure and brain chemistry that are occurring, and that's sort of the question that I wanted to seek out in the work that I've been doing so far. And I think this is actually critically important and sort of really needs to be married with the work that Dr. Sege is doing and others in the field, because with mechanistic understanding of why resilience can be brought about, we could build better diagnostic tools. We could understand the children who are particularly vulnerable, because what you might have appreciated even on that first graph from the Turner House study is that there's a wide variation in outcomes. We can also build new repertoires of treatments. We can mobilize interventions in a really evidence-based way. And we can really think about how our role as scientists can help inform the political discourse on really the critical need of social support systems. So how do we do this? How do we understand actually what's going on in the brain? Human studies are incredibly powerful. I love them. They form the basis of what I do. I think that for me as a molecular biologist, what I'm really interested in is the mechanism, and unfortunately human studies are limited in that. So what I would argue, as Dr. Lim sort of beautifully showed, is that we need animal studies. We need animal studies to build in mechanisms. We can control exposures, and then we can study what the effect of those exposures are. So if we're interested in resilience and positive environments, how do we do that in the mouse sort of setting? And thankfully, the field has already kind of answered this question for us. It's long been known that environmental enrichment of mice has a beneficial effect. So what I've put up here are actual mice cages that we sort of rear in the lab. And I should mention before I go on further, this work was done in very close collaboration with a former grad student in the lab, Scarlett Barker, and I'm really grateful for the collaboration and all of the really cool work that came from this. So this is standardized housed animals. So these are how we normally house animals in a laboratory setting, just with a little nesting and bedding material in a cage. What we can do is we can soup up that environment. We can make it enriched. We can provide a running wheel, which you can see that multiple mice are fighting over using. We can provide toys and mazes, more bedding for them to burrow, and we change these toys out to kind of create a more positively stimulating environment. And the perspective's off here, but I hope you can appreciate this cage is about 3 times as big as this, so they have more space to roam and burrow. And the field has actually already studied this extensively and shown that it actually stimulates and has an effect on multiple brain regions. And people have shown in multiple mouse models of different neuropathologies, whether it's neurodegeneration or stroke or seizures or traumatic brain injury, that mice that are exposed to enriched environments have better outcomes than mice are not, than mice that are not. So we already know this, and what we were interested in understanding is getting under the hood and looking at the molecular mechanisms that dictate this importance. So before we proceeded, we need to establish that enriched mice actually look enriched in our hands. And so what we did was we took mice during adolescence and exposed them to 1 month of this enriched environment and compared everything to the standardized housed animals. You'll see EE here refer to enriched environment, and standard housing are control groups. And we needed an ability to test whether they showed a behavioral effect of being exposed to that enrichment. So I'm going to walk you through a fear conditioning and extinction paradigm that we often use as a marker of learning and cognitive function. So in this behavior test, mice are exposed to a chamber in which they're given a mild shock. And so then on a subsequent day, we can ask, do they remember that this environment is now aversive? When mice are fearful, they sort of as an evolutionarily adaptive response, they tend to freeze. So we can measure their level of freezing and use that as a proxy of whether they learn having been shocked in that environment. We can also then couple this with a fear extinction learning test. So in this test, what we do is we actually re-expose mice to the chamber without a foot shock. So now they should actually learn that this environment is no longer fearful and start to freeze less if they're learning and able to learn that that environment is now no longer harmful. So when we exposed mice to a month of enrichment and checked their fear conditioning, what we saw was what the literature in the field already knows, but we were happy to replicate that mice that are enriched show elevated freezing after being shocked in a chamber, suggesting that they had higher and increased memory and learning of being in that environment. When we then looked at their ability to extinguish that memory, we saw that they were increased in their ability to extinguish that memory or shown enhanced fear extinction learning. So here you can compare the initial amount of freezing that they had after being shocked with their 5th day of testing and see that the slope there compared to the control animals was sharper, suggesting that they exhibit elevated and increased fear extinction learning. So great, in our hands we've shown that enrichment leads to cognitive benefit. So what are the key questions? First, what are the brain regions that are responsible for this effect? And second, how do we actually measure what's going on? So the first, we can rely on the literature. The field has long worked out the networks that are important for fear conditioning learning. We know that key regions like the prefrontal cortex, the amygdala, the hippocampus are important. And these are actually well dissected and studied in mice, and we know the same regions are actually important. So we chose to focus on the prefrontal cortex. Now, how do we measure what's changing? So there are actually some world-leading experts in epigenetics here, like my own mentor, Professor Tsai, and even Kate Pena. But in order to get us all on the same page here, I want to just give a brief primer in epigenetics. So if you can humor me for a second, if you imagine that the 20,000+ genes that we have, that we inherit from our mother and father, make up our genome, if you could imagine that those are libraries in a book, you could imagine that every cell in your body actually has the same genomic material. But what makes a brain cell and a skin cell a skin cell? The genes that need to be expressed in those different tissues are incredibly different. And so historically, what people found and how the field of epigenetics was discovered is that chemical changes to this genome actually lead to changes in which books are open or genes that are expressed and which books are silent or genes that are turned off. And more recently, we know actually now that the environment is actually a strong mediator of these epigenetic changes. So we actually know that the environment can actually lead to these chemical changes that affect how well expressed genes are in particular cells. What does this actually look like at a molecular level? So your genomic material or your DNA is a series of chemical sort of molecules that are wrapped around proteins known as histones, and actual chemical modifications to those DNA or those histone proteins can affect whether the DNA is open or closed. Now, our body also sort of synthesizes important proteins known as transcription factors. These transcription factors are important for coordinating the expression of particular genes. So we know if a particular area of DNA is open, then these transcription factors will be more readily able to bind to that DNA and express the genes. Whereas if the DNA is, the chemical modifications or the epigenetic changes have closed that DNA, that same protein can't bind there, and so those genes are not going to be expressed. And so this, in a nutshell, is kind of epigenetics. And recent advances in the field have allowed us to look at this epigenetic state of the DNA across the entire genome. I'm not going to go into the specifics of this methodology, but we can basically leverage different enzymes and different known sequences of DNA, transposable elements, to actually assay across the entire genome which regions of the DNA are open and closed. And so when we do that, this is kind of what the readout looks like. You can see in open chromatin that we're able to sort of identify more insertions, but in the closed chromatin, we can't. And this is actual data from a sample track of the genome from an enriched mice and a standardized house mice. And I hope you can appreciate that this area of the DNA or the genome is way more open than this. So when we did this in our enriched mice, I want to come back, it took a little bit of a detour. When we did this in our enriched mice and our control mice, we were really excited to see that in both males and females, there was an increased openness of the DNA after enrichment. So here each row is a different individual animal, and the columns are different genomic loci, and the heat map suggests yellow, brighter, or more open. So you can see that in both males and females, there are a larger percentage of genomic areas of the DNA that are opened after exposure to enrichment. We were also excited to learn that actually across both sexes, there's actually conservation of where those genomic loci are becoming more open. So what do we do with this knowledge? We actually know these locations in the genome. And if you go back to my analogy earlier of knowing that we know that transcription factors bind to the DNA, we can actually say, okay, what transcription factors, what proteins are binding to these regions that are now open after enrichment? And when we did that, we were really excited to see a class of transcription factors known as the MEF2 class of transcription factors come up as being enriched for binding into those peaks that are now open after enriched mice. The reason this excited us a lot is a couple. One, we can rely on the backs of the field because MEF2 is really well studied in neurodevelopment. We know it's a critical, important transcription factor family for neuronal health. But it's been a little bit less well studied in the context of environment and how environment might be affecting it. And so that led us to want to study it more. The second is it's actually been implicated in a lot of genetic studies. We know that children that have mutations in MEF2C particularly suffer from intellectual disability, and there are actually GWAS studies or genome-wide association studies that show MEF2C is associated with modulating intelligence. So we first wanted to validate this. We looked in our mouse brains. Dr. Lim talked about immunofluorescence. So we stained for MEF2 levels in enriched and standardized housed animals and found that there was increased MEF2 in the brain of animals that were enriched. And so now if we take our simplified presumptive model of good environments being good because they're good, we can actually break it down into mechanism. We can say that enrichment is leading to an increased MEF2. But what we want to prove is that is this causal? Can we put this in the link between why a good environment is having benefit? And so to do that, we rely on another technique. I really put Dr. Sege and Dr. Lim before me as plants because Dr. Lim beautifully introduced shRNA. We can actually use this technology to knock down proteins in the brain, as he showed. And so we did that to knock down the MEF2 family members. And then we exposed these mice to an enriched cage. So now if MEF2 is actually important for affecting the cognitive benefit that's resulting from an enriched environment, we should be able to block the effect or the cognitive benefit that we saw. So this is another immunofluorescence staining so that we were effectively able to reduce the levels of MEF2 in the brain. And we returned to our fear extinction learning paradigm. And what we were really excited to see was that when we use a control shRNA, which doesn't knock down any protein, we still see that really nice fear extinction learning in the animals that are exposed to enrichment. But when we add the MEF2A and C, when we knock down the MEF2 family members, we actually impair that extinction learning, causally implicating MEF2 as an important factor in the pathway of translating enriched environment to cognitive benefit. So I am a physician. I do all this mouse work because I think it's actually really important for what's going on in humans, but can we prove that? So we can actually turn to the published data in the field, because about 10 years or so ago, or I guess it's now longer, but 10 years ago they published gene expression analysis from this study. But there's a study that's run by Dr. Bennett out of Rush University in Chicago, known as the Religious Order Study and Memory Aging Project. And what they did, starting in the 90s, I believe, is enroll adults longitudinally. They performed cognitive testing on them serially, and when they passed away, their brains were donated to science. And so of 600 of these individuals, they performed gene expression analysis on brain samples. And we actually know, because they were tested while alive, what their cognition was right up until the point that they passed. And so we can really sort of marry gene expression data with clinical and phenotypic exposures. And so as a sanity check with these patients, the first thing we wanted to do is test, like in our mice, does enrichment actually lead to cognitive benefit? And so what you see on the x-axis here is lifetime cognitive activity score. They had a validated survey where they assayed how mentally active individuals were on a daily basis. And then here you have their cognitive testing, which was actually done by a clinician in the field. And they saw a significant association between the more active an individual was, the more likely their cognition was to be higher. Each individual point here is an individual from the study. And so what we did was we wanted to say, okay, does cognition correlate with the levels of MEF2? And what we saw was that there was a very significant and positive association between cognition and MEF2C in the gene expression analysis from the brains of these patients. So the higher an individual's cognition was, further on the x-axis, the more likely they were to have higher MEF2C expression. So while this is still just kind of correlational, I think married with our mouse studies really nicely kind of pinpoints the role and the critical importance of MEF2 transcription factors in mediating cognitive health. And so in the interest of time, I just want to gloss over this a little bit. The brain is really complicated. It's multiple cells. You have neurons, you have vessels, you have glial cells that form like the myelin and do a lot of different things. I don't want to summarize this, it'll take a while. But we can harness now new advances in technology that are known as single nuclear RNA sequencing to, at a single cell level, determine where MEF2 is active in these resilient patients. And I'm really excited. This is really a powerhouse effort between Professor Tsai and Professor Manolis-Kellis' lab, where they now have collated over almost 500 subjects where we have individual single cell data on these patients, and we can track gene expression correlates of cognition. So returning to the working model, it's really quite simple, but it took a lot of work. We can translate environmental enrichment into increased MEF2 activity, and that this MEF2 activity is critical for increased cognitive health and cognitive function. So in future directions, we want to be able to test whether this could actually help promote and resilience against neural insults. We actually already have data in the field, in the lab, that shows that MEF2 is able to rescue the cognitive neurodegeneration in Alzheimer's disease mouse models, but we're going to expand that work. We also want to screen for activators, because if you could somehow find a way to activate MEF2, then could that be a way and a tool to improve cognitive health? And interestingly, I don't know where to put this, but I'm just so excited about this. People have looked, and it's been known since 2001 that in muscle cells, activity and exercise induces MEF2. So could we also be sort of harboring and narrowing in on the mechanism through which physical activity is so important for brain health? And could that be MEF2 dependent? In the last minute, I've talked a lot about enrichment and our models here, but as I started with ACEs, I'm also very deeply passionate in understanding how stress, and early life stress in particular, affects brain development. And we have a couple of other projects in the lab where we're looking at how chronic resource deprivation affects gene expression in the epigenetic state of the brain, and how social isolation affects gene expression in the brain. I think this project is one that I'm particularly and keenly excited about in the wake of what is a growing and I think burgeoning pandemic of mental health disorder in the wake of the COVID-19 pandemic. In 2 years alone, there's been a 4-fold increase in the rates of reported anxiety and depression, and that's reported. So we know that these disorders are largely underreported, and this has been largely attributed primarily to the multifactorial effects of the COVID-19 pandemic. This is a global phenomenon. So in almost every country they've looked, there's been at least a 2x increase in the amounts of mental health disorder that people or patients are reporting. And this is a complex landscape, as I just alluded to. The secondary effects of COVID-19 are immense and massive. How do we start to break that down people across their various fields here are going to be doing their kind of respective work in these areas. As neuroscientists, we're keenly interested in how the mental health impacts of the COVID-19 pandemic are affecting outcomes. And I found this study from the Harvard Crimson actually to be really interesting. So they surveyed they got about 80% of the class of 2025 to respond to the survey. So this was in 2021 when they were enrolling in freshman year. And 63% of incoming freshmen reported a decline in their mental health, which they could attribute to the pandemic. And the leading cause of what was causing that decline in mental health was social isolation. We're going to be inundated with studies like this one out of Uruguay, I think, in the coming years, because now we can retrospectively go back and look at what COVID did. And what these researchers did was they had looked at children and their cognitive development from ages 4 to 6 before the pandemic, and then during the pandemic, and matched patients for all other variables like socioeconomic status and ethnicity, and found that there was a significant hit that cognitive and motor development took during the COVID-19 pandemic. So that framework that I just showed you for enrichment, we're now leveraging it to look at social isolation, and we're really excited to kind of harness the power of human gene expression data sets, marry that with mouse studies to look at mechanism, and really identify the conserved changes that occur in response to isolation in both humans and mice. I don't have time to get into the data here, still pretty preliminary, but happy to chat about this offline. So with that, I'd like to stop. This work was only possible because of the mentorship of Professor Tsai and the team that I work with, and I mentioned Scarlett Barker there. Also really thankful for collaborators and my clinical mentors. And really, I want to take a moment here to thank the JPB Foundation. I was a recipient of a clinical fellowship when I was a resident, and that's really what protected my time to be able to do this research. And for those of you who trained in medicine, protected time in residency and fellowship is really rare, and I feel really fortunate to have been awarded that. So with that, I'd like to stop and take any questions. Yeah. Oh, I can repeat the question. Yeah.
Sorry.
Oh.
Does the age at which enrichment is provided affect efficacy?
Yeah, we've done a few ages. We've done adolescence. We've also done adulthood, as I alluded to in our Alzheimer's disease models, and we've shown benefit at both. And the studies that I highlighted in that review have done all ages. That's really a great review to turn to for an overall summary of the field.
Hi, Ravi. Very nice talk, very important work. So have you happened to look at adults with PTSD? Do they have changes in MEF2 expression levels?
Yeah, I'm so interested in expanding this out to other neuropathologies. The ROSMAP data did not look at PTSD as an input variable or outcome variable, so we don't know that, but I'm sure there are some human databases of PTSD that we could look at. I've become a huge fan, as Leeway will know. I think the field has so much data, and I think you can do so many experiments just like sitting at your desk on the computer. So all these data sets are publicly available, so we can go now and look and ask and sort of determine. I mean, it might take a few weeks or months, but we could eventually get there. So it would be cool to look at. Yeah.
Okay, one last question.
Yeah, if you think about the population of people with those 3 or 4 or greater adverse childhood events, there's a lot of them, a significant fraction of them that have some sort of mental health issue, but there's also a big population that don't. Is there anything that we could learn from that population that don't? Is there something about their genetic or environmental profile that puts them back on that trajectory that you mentioned that we could model as a treatment or something for the other members of those groups?
Yeah, I love that question. I think, and it kind of is a nice sort of, especially after coming Dr. Sege's talk, but, oh yeah, thank you, sorry. So the question was, can we learn about the people that are actually resilient to stress, if I'm understanding correctly, and then why they're resilient to that stress in terms of endogenous mechanisms, as opposed to why the environment is beneficial, potentially. And I think that's really cool. I had an earlier project that I eventually abandoned because of the variability, where we were trying to differentiate mice that were stress exposed into resilient and vulnerable. And I just, in my hands, had a really hard time sort of making that a reproducible sort of phenotype. But there are people in the field who have managed that, and I would love to sort of learn from those studies or try to sort of launch them myself.